Good morning and welcome to the Centers of Excellence in Iron and Heme Disorders at the University of Utah for our series Interviews with Giants in Iron. Today we're privileged to have Dr. Jerry Kaplan speak with us. Jerry got his bachelor's degree from Stony Brook in New York. He then went on to get his PhD at Purdue. There we go. There's the hat. Awesome. Do you have the Purdue hat too? Oh, way to go, Jer. Anyway, got his PhD at Purdue, and then he went on to a postdoctoral fellowship at Harvard. And his first faculty position was at the University of Connecticut. And from Connecticut, he was recruited to Utah by Ray Danes, I think. Is that right, Jer?
Right.
Okay. And started in the pathology department here. And today we're privileged to be able to interview him and Cade Loveridge, who is an MD PhD student here at the University of Utah, will be conducting the interview. So thanks so much, Jerry, for taking the time to do this. And go for it, Cade.
Awesome. Well, I'm also very excited for this interview. I'm grateful to be a part of it. If you wouldn't mind just kind of giving us a background of you and your family, that'd be a great way to start.
Sure. I was brought up in Brooklyn, New York. I am the second generation citizen. I hate that term. I was a second generation born in the United States. My parents were lower middle class. My father worked as a salesman in a haberdashery store. My mother had various jobs as an accountant. I went to public schools, and actually public schools, public colleges. The only time I went to a private institution was a postdoc at Harvard Med School. So my parents were not college educated. My father was brought up during the Depression, and there were times when he was the only person in his family who had a job. So he graduated from Knight High School. He started to go to Knight College, and then the war started, World War II. Got out, was married with a kid, so he started working. So in my family, there was never a question of whether you wanted to go to college, is you're going to college. And in fact, that whole generation of Jewish kids, the same issue held. There was no question of whether we were going to college. Everybody went to college, except one guy, when he graduated high school, went to the racetrack. But
is he the rich guy?
I don't know what happened to him. Probably not. Right. So everybody in my generation got educated, curiously enough, that all the women, and even in the younger generation above me, my mother's younger cousins, all the women who went to college became teachers. And a lot of the men, with one exception, one woman did not become a teacher. She became a lawyer, and then a judge, and then a judge, and sat on the Supreme Court. But the rest of the women they thought being a teacher was perfect for them. The men did different things, and I was the only one who ever became a scientist.
So that's a perfect segue into, kind of could you give us a broad overview of your scientific career?
Sure.
How you got into science and where that, led you throughout the whole process?
Sure. So I went to college as a biology major, and because I always liked fishing, and I lived actually on the coast of Brooklyn, I expected to be a marine biologist. But I had an NSF summer grant. No, I had an NSF grant during the spring semester of my sophomore year, and we were sampling plankton, and we went out on the Long Island Sound in an open boat in winter. And when the hair in my nose froze, I knew I should do something else. Right?
That's fair.
But I've always been interested as a kid in the coastline, right? The boundary between two worlds, right? The ocean and the land. And in science, I always got interested in plasma membranes, right? The boundary between two worlds. And in one summer, I had another fellowship where I studied sponge cell adhesion, in part because I could die from my own sponges. And that's a very famous issue in developmental biology, where you can take two sponges, squeeze them, a red one and a yellow one, squeeze them through cheesecloth. The cells will then fall apart. They'll hit the bottom of a flask or a petri dish. They'll move around and they'll reform a red sponge or a yellow sponge. So how did the cells readhere and how could they know who's who? So that interested me. I got into college at Purdue solely because of that work, certainly not because of my academic grades, by a guy a faculty member, Merlin Moskowitz, who was also interested in cell adhesion studies. And I studied membrane turnover. When I graduated, I did a postdoc in animal virology because I thought using animal viruses who bud off from the cell surface was a great way of studying membrane biogenesis. It was a very good idea, and I think probably 60% of all the young scientists in America came up with the same idea. So I did that rather non-productively for a while, and I went back to studying membrane turnover, and the cells I chose to use purely by serendipity were alveolar macrophages, a very highly phagocytic cell important for lung protection. So I then started studying membrane proteins on the surface of macrophages, and I was heavily invested in endocytosis. And it turns out that a lot of people who were invested in endocytosis, Harvey Lodish, and it'll come to me in a minute, went into the transferrin receptor. And the transferrin receptor was really noted for being internalized, although at that time where it was internalized was not known. So I got interested in the transferrin receptor. So Diane told you that I was recruited to Utah, and I was by Ray Daines, who was in pathology. But in those days, they would run me around and see other faculty. And because I was interested in the transferrin receptor and I hadn't published on it, I asked to speak to George Cartwright, who was the head of medicine at the time. Utah got famous in internal medicine. For many years, Utah was one of the top places in America for internal medicine due to the activities of a guy named Max Wintrow. The Wintrow building. Max Wintrow was actually an MD, PhD, although there were separate degrees at the time, at Johns Hopkins. And he was one of the world's most famous hematologists. But in the 1930s, the president of Johns Hopkins came to Wintrow and said, you know, Max, Dr. Wintrow, because you're Jewish, you will never be promoted to a full professor at Johns Hopkins, which is a great quote. He got recruited to come to Utah, which was a 2-year medical school. So he had never seen it, came out here, found himself in a desert, but stayed. And he oversaw the transition from a 2-year medical school to a 4-year medical school. And he hired wonderful faculty members. And this place became really hot. And George Cartwright was his accolade, right? He was acolyte. And George became head of medicine. And George, and we'll get into that later, had a real interest in diseases of iron, and transferrin is the iron carrier. So I started speaking to George Cartwright, and coming to Utah because of Cartwright became more intriguing, although it wasn't the major reason I came here. So I've just corrected some revisionist history where people in hematology for a while took credit on recruiting me here.
So where's the Kushner story come in there?
All right, this is a great story. Kushner, James Kushner, James P. Kushner, right? Was a faculty member in hematology. And I would say that, and for reasons that I'll describe later, I call him Kushner and he calls me Kaplan. It's a tradition for people in New York to call each other by their last names. Kushner really was a solid member of hematology, a great scientist studying porphyrias. When I met him, he was an associate professor, and I had accepted to come to Utah. And after I accepted in February, but in April, George Cartwright died. No, I'd only met him twice. But one day out of the blue, I get a phone call from a guy named Kushner. He says, my name is Jim Kushner. I'm a faculty member in hematology. George Cartwright wrote an NIH. George Cartwright was NIH funded all his life, but he had written an NIH grant, which was funded, in which half of the work had already been done. And the other half to be done, he was going to do with me, although he never told me that. So Kushner said, I'm coming to New York. Perhaps we can get together and talk about, if we're simpatico, we can work on the grant together. And I said, sure. And I'm living in Connecticut. We decided to meet on the steps of the Plaza Hotel. And I said, well, how will I recognize you? And he said, I'll be the guy with the payas. Now, the really Orthodox Jews never cut their sideburns. They're long and dangling. And in Yiddish or Hebrew, they're called payas. And I said, okay, hung up the phone. I said, he's not an Orthodox Jew, right? So I spoke to a colleague of mine at Connecticut who had been a fellow with him. And I said, is Kushner an Orthodox Jew? And he said, oh, no, not at all. Okay, the guy's blowing smoke at me. So I went to a theatrical supply store and I bought a wig with a beard and payas. And I stood on the steps wearing this, but how will I recognize Kushner? So I had a sport jacket, and in it, I had a copy of the New England Journal of Medicine opened up to an article he had published. And that's how we met. And as a present, I gave him the wig. And we became friends. And we took up the grant, and we worked for all of our time together in Utah, which is 40 years. He was head of hematology oncology, head of the clinical research center. He was one of the smartest guys I've ever met. He had an eidetic memory. He never forgot anything. He was an absolutely clear thinking. And I think he was one of the best diagnosticians in the Western United States. He was great.
100%.
Yeah, he was wonderful.
That is a amazing story. I'm glad that that came up.
Yeah.
And so that kind of paints a good picture of, kind of how you came to Utah, how you got involved in iron metabolism research. From transferrin, can you kind of give us an overview of, like a brief synopsis of your lab and kind of some of the different problems you helped tackle and understand?
Sure. So when I came to Utah, I had never worked on iron and transferrin. We had studied endocytosis. So the first paper I ever published on transferrin, uh, ask Diane who the first author was.
My brother-in-law, John Ward.
John Ward, who was a young fellow in HEMONC and joined my lab. I think he was assigned to me by Jim Kushner. But we started working on transferrin together. And the first couple of papers ever published was by John. And you can ask Diane how she met her husband later, right?
Yes, you
He
can.
was John's brother-in-law. Anyway, so we started studying on transferrin, and we actually had a hypothesis. Do you know anything about the disease hereditary hemochromatosis?
Mm-hmm.
Okay. So your audience probably knows, but it's a genetic disease that results in iron overload in most of the parenchymal tissues of the body, liver, heart, pancreas. It's also hemochromatosis really means bronze diabetes, right? Gives rise to arthritis. And that had been a long-term interest in George Cartwright. So I'm going to digress for a minute because I don't know if this came up. George Cartwright, so a French group discovered that there was a genetic association between hemochromatosis and an HLA antigen. And George Cartwright, who was such a forward thinker, hired a faculty member who had a background in genetics to study this phenomena, to localize the hemochromatosis gene. That guy was Mark Skolnick, who studied myriad genetics and was responsible for cloning the breast cancer gene. But Mark first started out studying hemochromatosis, and to identify this association, you had to use complicated immunological tests to define the HLA antigen. So 40, 50 years ago, he brought up this problem at a meeting of the genetic training grant. The genetic training grant always held a meeting 1 year at the Alta Lodge. It was actually pretty famous. And 1 year they invited a scientist, a yeast guy from MIT named David Botstein. And sitting around drinking bourbon, they try to figure out, is there a better way to localize genes? And Botstein came up with restriction fragment length polymorphism, using differences in the DNA sequence and enzymes that cut sequence with sequence specificity to localize DNA fragments close to a phenotype. And that led to the first genetic map, and that led to the discovery of diseases because their phenotype was associated with the genotype. They wrote a fantastic paper on it, which had 4 authors: Botstein, White, Skolnick, and Davis. I may have gotten the order wrong, right? 3 of the 4 guys are in the National Academy, and the 4th is a multimillionaire. But because there are 4 people on the paper, they weren't permitted to be considered for a Nobel Prize, which limits the prize to 3. But that was one of the papers that made Utah really the genetic hub for America for 30 years, all because of an interest in iron.
That's pretty amazing.
Right. So when I came here, we still worked on endocytosis, but we started working on the transferrin receptor because I had a hypothesis that unregulated activity of the transferrin receptor uh, could lead to hemochromatosis. But in John Ward's paper, the first thing he showed was that transferrin receptor number is regulated, and it's regulated at the level of synthesis by iron. And in another paper, he showed that transferrin receptors on cells, fibroblasts from people with hereditary hemochromatosis are perfectly normal. So that led us to the conclusion that hereditary hemochromatosis is not due to conventional transferrin receptors bringing in iron. At about the same time, someone gave me a present. The present was a mouse that did not have transferrin. And this mouse, as you might expect, exhibited severe anemia, but yet at the same time had iron overload disease in all of its organs. The name of the guy was Selden Bernstein, and Selden was a faculty member at Jackson Labs and was really responsible for finding a variety of other mouse genetic disorders having to do with red cells. So he found this pale little mouse, which promptly died, but he found that if he injected it with mouse serum, he could keep it alive, and that if he injected it with purified transferrin, he could keep it alive. Well, it turns out at that time, there were 3 human patients that had apotransferrinemia, and one of them was kept alive by Boehringer Ingelheim. It's a big pharmaceutical company. They're in the United States, they're in Germany, but the 2 companies were split. And that company provided the young girl with purified transferrin, not serum. So she actually escaped the AIDS epidemic. But that mouse and those patients took me to a series of papers done by 2 Army guys at the time, William Crosby and Muncie Wiebe. And what they showed is that you don't really need transferrin to absorb iron from the gut. And that in patients with hemochromatosis, I think they did this experiment, they absorb iron even though their transferrin is completely saturated. And we reproduced their experiments in the apotransferrinemic mouse and normal mouse. If you saturate transferrin with iron by injecting animals with iron, and then either inject them or give them a radiolabeled dose of iron, all of that iron goes to the liver and the pancreas and the heart. So that told us you don't need transferrin to absorb iron, and that there is a transferrin iron transport system, and a transferrin independent iron transport system in a variety of tissues. So I'll stop there, or I could go on.
No, that's perfect. Keep going.
I think you should go how you hopped into yeast, Jer.
Okay. So there's a transferrin independent iron system. We did a couple of experiments in cultured cells showing that cells would take up iron, but we didn't know how to go after that system. So, we're thinking of different ways, but it hit me that I don't know anything about plants. And it was a rainy spring Saturday morning, and I went down to the university library because the medical school library doesn't have very much on plants. And I'm starting to go through the plant literature. And I came across a paper and I looked at the title of the paper and I read the abstract and I literally started screaming. In fact, the librarian told me to shut up. It was a paper in which these authors, Welch, give me a second because I wrote it down because I want to do them. You can see how organized I am.
It's one of my favorite things to remember how we used to go to the library and go through the stacks. I mean, you'd find a paper that you needed to read, and you actually went to the library and you like sorted through all the old stacks and journals to find the article, pulled it out, went with a Xerox card and made a copy of the article so you could have a copy.
Did you tell Cade Spangrud's rule?
No.
There was a graduate student named Jerry Spangrud who came out of the library one day and he said, I just made a discovery. 6 months in the lab can save you 5 minutes in the library. And of course, he was being ironic, right? Yeah. So I love going into the library. Anyway, I found these series of papers, one of the, in which they described the mutant P. So the wild type P is sparkle, the mutant P is bronze, and the mutant is a sickly little plant. And it turns out it hyperabsorbs iron, puts the iron in the leaves, and the iron and gets brown spots, necrotic spots from the iron. So in the paper, the authors describe that they named the mutant bronze because of the bronzing of the leaves. But in the discussion, I think these people called it bronze because they said the phenotype of this plant looks like the phenotype of human hemochromatosis, bronze diabetes. You hyperabsorb iron in the gut and you stick it in the liver and the other organs. Excuse me. So I said, if plants and humans can show the same disease phenotype, maybe they use the same strategy. What's the simplest multicellular organism? Yeast. So I became a yeast geneticist. And in doing so, we were really lucky because I got a postdoc at that time, a guy named David CIHD, who was he just came off a 5-year postdoc in yeast. So this guy knew yeast backwards and forwards. To me, as a cell biologist, tissue culturist, yeast were those cells that contaminated my tissue culture. So David was a great help in the lab. And I gave David a technician, a woman of great virtue, my wife, and who has spectacular hands. And if she did an experiment twice over 20 years, it would get the same result. Probably because she kept the same reagents. But she was great. So with David, we did our first set of experiments on yeast. And they led us the result that there was a high affinity ion transport and a low affinity ion transport, and they were separable. Right? So based on that, we set up a genetic screen using a reagent discovered to be of use in bacteria called streptonigrin. Streptonigrin combines with iron, generates an oxygen radical, and kills cells. So you take yeast, you put them on, you iron starve them, which induces the high affinity iron transport system. You give them iron, you give them streptonigrin, and it's just a pulse, and they die. You now take away the streptonigrin, feed back iron, and yeast survive. But the mutants are the ones that died were the ones that had lost the high affinity iron transport system, but could be rescued by high iron in the lower. So that gave us a whole bunch of mutants. And the first of those mutants was we had a whole bunch of mutants, but I'm trying to remember why we focused on FET3. I mean, there was a FET1 and a FET2, but we focused on FET3. And FET3 was a really interesting mutant. And as I tell people, FET3 helped send my first two kids to college. It encoded a protein that is very, very homologous to ceruloplasmin. It has many coppers, and it's a copper oxidase. And what it does is it takes reduced iron, Fe2, turns it to Fe3, and in humans, ceruloplasmin puts it on transferrin, which takes it to cells. In yeast, FET3 takes iron and puts it on FTR1, which is a membrane transporter, which then brings iron into cells, right? So we set up this mutant screen. We had a mutant. We then put in plasmids, genomic plasmids, and looked for guys that can complement the mutant and hoped that the normal complement of the defective gene was on the plasmid. And this was done by a young graduate student named Candace Asquith. Now, we didn't know how to sequence. We've never sequenced a gene. But our colleagues, in this case, not Ray White, Ray Jesslin, had a big sequencing lab. And so they took her under their wing. And she came back and she said, the guys had me sequence just a little part of the plasmid. And as an exercise, we put it in the database, and they got they got a hit, they said. And I said, what is it? She said, I don't know, something called multi-copper oxidase. And I screamed again because I knew what a multi-copper oxidase was, because the role of ceruloplasmin in iron transport had been worked on by George Cartwright. Turns out they studied copper and diseases of copper in pigs. And you make pigs copper deficient and they become anemic. And they become anemic because they don't have ceruloplasmin. So we're very excited. Candace goes back, sequences the rest of the gene, and they use the very unique sequencing procedure, which works, but is not commonly used now, in which you do a lot of prep work, push a button after all of this prep work, and you sequence 10,000 bases at a time. So Candace is doing that, and now she's doing it right before she goes back to vacation. And Candace was a graduate of the University of Michigan, so she was going home to Michigan. My son at the time was going back for his sophomore year at Michigan. He was an undergraduate. They were on different planes, but at the same time. So I said, Candace, I'll pick you up and take you to the airport. So there she is, sitting on the sidewalk in front of her house, suitcase in one hand, and a readout of 10,000 base pairs in the other. They had sequenced, they had pushed the button last night, and we got the sequence. So that resulted in us finding FET3 and also establishing, again, a role for copper in iron metabolism. That was great. And we did a lot of things with FET3, and we found a lot of other interesting mutants as well.
Yeah, that's you're a very good storyteller. It's amazing to hear about literature that I've been reading for a long time and kind of hearing the process and like the logical steps that got there. So that's really cool. I know you've mentioned a lot of colleagues and trainees that you've worked with and benefited from. Are there any other people that come to mind that you'd like to shout out?
Well, sure. So in the end, well, first of all, Diane. Diane has been with me
40 years.
40 years, right? She started off. So, truth be told, I come to Utah, and in my first year here, I get invited to talk at BYU. And so I was nervous. So I took John Ward with me, right?
Because you have to give the logic, because he was Mormon and you were going to BYU, and a Jew at a Mormon school was a scary thing.
It was, but 50 years, let's go 50 years from that. What is the religion of BYU's star quarterback?
Exactly. That's exactly what I was thinking. Yeah.
But I was, to be honest, nervous. Right?
Yep.
So I took John with me, and we had a great time, right? And we're in a room talking to someone, and this young woman walks in, although she didn't talk to us, and that was Diane. So I think at the end of the year, Diane graduated from BYU, applied for a job, and she came into the lab as a technician. And Diane can do anything. I mean, right? From I was about to say from training mice. I mean, for really working with mice to cultured cells to yeast. But as I said that, I remember this story. We were doing something with mice in a hood, and I'm on Diane's right side, and the mice escaped towards the right, and Diane literally picked me up and moved me away so she could capture the mouse.
He wasn't moving fast enough, and the mouse was going to climb up the back of the fume hood.
Yeah.
So he had to get out of the way.
Yeah. And then I had gone downhill skiing once before in my life. And it was in Laurentians, which is north of Montreal. And I had a ski instructor for 30 minutes who did not speak English well. I did not speak French at all. It was a T-bar lift, which is frightening even today, and you're skiing on ice. So it was a disastrous experience. But I'm in Utah. I've been here for a couple of years. It was, I think, just before my 40th birthday, and I felt morally obligated to go skiing. Now, it turns out I had cross-country skied. So I went skiing. So what did I do? I took Diane, who was an excellent skier, and I took John, who's an MD. So it turns out I did okay.
You did very well.
Thank you.
No problem. Your cross-country skiing made it so you had no problem.
Yeah.
Yeah.
So I had Diane all these years. The other I had other people. Rich Agioka worked on, and he actually worked on endocytosis and transferrin, but not on iron delivery. We found a way that we could hook up an enzyme to transferrin, watch it get internalized, put the cells at 0 degrees, and turn the compartment that transferrin was into cement. And then ask the question, are all the transferrin receptors in that compartment? Are other ligands in that compartment. And it was very useful for mapping receptor and endocytic behavior. But the other guy who deserves a lot of note was Liang Tao Li. Liang Tao Li. Liang Tao was my age, had been a physician in China and got chewed up by Mao Zedong and had been sent to the countryside. He came to Utah because he had a brother who was a pharmacist here, and Liang Tao was starting to go was to clinical lab medicine, to be a clinical lab tech. And he had no money, and he got a job here working part-time. And he initially started working on mice. Turns out Liang Tao also could do anything.
Best
He
cloner in our lab ever.
He could actually make, we could do timed experiments with mouse urine because he could make the mice pee on command, right? But he also turned out to be an exquisite yeast geneticist.
Yeah.
Yeah. So we had Liang Tao taught by David Eide for a year. And David Eide went on to have a great career going from iron in our lab to zinc. And David also cloned the low affinity iron transporter, and he devised a yeast cell that had no high affinity iron transport. And that was used to great effect by Mary Lou Guarino and others to clone plant genes that encode iron transporters. And we did the same thing with the lysosome. I don't know if I'll get to that. Liang Tao was spectacular. I had Diane Liang Tao, and then I had 3 great graduate students. At the same time, Candace, who worked on iron in PhD3, Derek Rudisky, who had been a chemist, who worked on a bunch of iron things, and Derek was absolutely fearless. He would make any chemical, he would read any paper. For journal clubs, he was reading a paper by a Nobel Prize winner, and he had no compunction about calling him up and said, "Would you explain this sentence?" Derek is wonderful. He's now head of cancer biology at Mayo Clinic, Jacksonville. And then the last guy was Chuck Perot, who had nothing to do with iron. Chuck was the last guy. Chuck worked on endocytosis, and he identified a gene for a human disease called Chiari-Higashi syndrome. And it's a great story, but will take too long for the iron community. But he did a wonderful job. And then I had a great postdoc called Deepika, who was just marvelous. Yeah.
A lot of
So
good.
I really had a great group. That was, and there was Sandy. Yeah. And the lab, by the way, all right, you're interested in science and not stories. Right?
Absolutely. So
Let's talk science. Yeah.
In another interview with
Another interviewer. After the interview, I'll tell you the story.
Yeah.
I have a little bit of a loaded question. I have 2 loaded questions for you next. The first one is, if you had to choose 1 or 2 of the many fantastic discoveries you made and your lab made over the course of your career, what those would be. And the second would be, if you had to choose 1 or 2 questions that still kind of keep you up at night about, iron metabolism in general, what would that be?
Sure. So yeast genetics was wonderful. I love yeast genetics. We discovered so much in yeast genetics, and I absolutely love designing genetic screens, right? And PhD3 was a great discovery. The other great discovery was a protein called YFH1, which is the homologue to human frataxin. A defect in frataxin leads to a disease, Friedreich ataxia. And we had discovered YFH1 through a genetic screen. We're working on it. And all of a sudden, we read in Science about this frataxin gene, and it's a yeast homologue. And we knew what frataxin did. And we knew how screwing up frataxin gives rise to pathology. And that, I think, was one of my biggest discoveries, because it could directly help people in non-subtle ways. And it showed that you could study diseases, even neurological diseases, in yeast if the genes are homologous. So that was great. I also discovered how iron metabolism in yeast is regulated. I did not discover AFT1, which is the transcription factor that regulates the low iron regulon, right? But I did discover that AFT1, the guy who discovered that was Rick Klausner, another early figure in the yeast iron field, and actually a really true wunderkind. And he was also a wonderful person to have as a colleague. So we were competitors and colleagues, and we published together. That's the other thing. I had wonderful colleagues in the yeast iron field. They were all gracious, and they were all generous, and I really still value them. So I understood how the low iron system was regulated. Well, we discovered there's actually a high iron regulation system, which involves storing iron in the vacuole, which is the yeast lysosome. So we discovered that. We discovered the transcription factor that regulates this, the transporter for lysosomal iron storage, called CCC1. And we also discovered how it's regulated. It's regulated by the same guys that regulate the low iron transport system. So if you don't make iron sulfur cluster proteins, which is made in the, all right, let me step back. The mitochondria has two major roles with respect to iron. You make heme, and you make iron sulfur clusters, as Roland Lill probably told you guys. If you don't make iron sulfur clusters in the mitochondria, you turn on the iron transport system. You turn off the high iron response. So the two responses are regulated reciprocally, which makes great sense. In human terms, the iron transport system, transferrin, is regulated inversely to the iron storage system, ferritin. So both in humans and yeast, the same factors that regulate iron uptake also regulate iron storage in the opposite direction. So I'm really proud of that, because we found that out by genetic screens. So the two things, the overriding factor is I'm still unclear on how you control the movement of iron within cells, right? What regulates iron, what prevents iron toxicity? And how do you move it from mitochondria to cytosol? So in yeast, we is really Liang Tao, discovered that there are two transporters that take iron out of the mitochondria. There are two transporters that take iron in, and two that take iron out. And in yeast, you can also store iron in the mitochondria. So if you get rid of CCC1, you can overexpress the iron transporters and store iron in the mitochondria in a non-toxic form. If you screw around with iron sulfur clusters, that iron becomes toxic. And that happens in humans as well.
We're on again. Okay, so you were talking about.
Yeah, so I love yeast. Now, in terms of importance, figuring out how hepcidin regulates ferroportin is really important, because at a moment you understood the phenotype of many of the genetic iron overload disease. Some diseases.
We were all screaming. We were all screaming.
Yeah. And actually, that's a great story. We started on this approach to, because I was interested in ferritin and how it stores iron and how it gets degraded. And everybody at that time, when they wanted to make cells iron deficient, they would use iron chelators, which I thought was really unphysiological. So ferroportin had been discovered, and we got the cDNA for ferroportin. I think her name was Adriana. I can't remember. It's my age.
Adriana.
What?
Adriana.
Yeah, Adriana. A very nice young woman who had discovered in yeast. For a while, I was the zebrafish people's iron yeast guy. So we had a long and really fruitful relationship, because if they discovered an iron-related gene in zebrafish, I would put it in yeast. And I had great collaborations with Lenzon and Barry Paw, who sadly passed away. Anyway, Adriana sent me the clone. And a medical student who had started in my lab as an undergraduate, and Marie Spencer, who worked that summer putting ferroportin in an expression clone with a GFP tag, doing one or two things, putting it into cells, and it was beautiful. The cells were green on the floor, had green fluorescence. It worked. Now, this will be interesting because you interviewed Tom Gantz. So I was giving a talk at UCLA, and I met Tom Gantz. And I knew of his work. I'd never met him before, although I had read his papers. And we're talking about iron and hepcidin and what could be the receptor for hepcidin. And I said, I just made this interesting construct. And why don't you send me some hepcidin? And he did. And I put it on cells, and whoop, the ferroportin on the cell surface disappeared, went inside, and Diane took the picture of that. Yeah. So Tom and I, for a number of years, had a very productive collaboration. Oh, but getting back, so the two things I'm really interested in, if I was to work again, is how is iron distributed between cytoplasm and mitochondria? And I think there's great evidence of how it gets out of lysosomes, or in yeast, how it gets out of vacuoles. The next thing is I'm interested in is we discovered two proteins involved in sterile biosynthesis. One of them is methylsterol oxidase, which is in yeast is called ERG29, and it's a great story about that. We had cloned the gene, Lingtown cloned it. Under low iron conditions, cells did not grow. And we actually tracked down why they didn't grow is they made funny sterols. But we knew nothing about sterile biochemistry, but we did thin layer chromatography, and we could see sterols we couldn't explain. So I then called up one of the world's experts on yeast sterols, a guy named Martin Bard, who was at a school in the university called Indiana. It was an amalgamation of Indiana and Purdue in Indianapolis. And I said, Dr. Bard, my name is Jerry Kaplan. Listen, we found this protein in yeast and we've cloned it. It has this funny phenotype. And he said, do me a favor, give me the first 10 amino acids. And I did. He then gave me the second 10. He had just cloned the same gene. And so we started talking. He sent me sterile intermediates. We published, I think the same month, we published in JBC and he published in PNS. And he had deliberately gone after this in a very sophisticated, way of identifying it. And we had a much simpler way. But for the next 15 years, we collaborated together, and he would do our sterile analysis and send us stuff. We would send him stuff. And I never met him. It took me 15 years to meet him. But we would talk usually on Saturday mornings. And our backgrounds were really quite similar. He was from New York. He had gone to Berkeley, went on and on. Yeah, he was great. He was a wonderful collaborator. All of my collaborators in the yeast field were wonderful, absolutely generous. When you asked for something, they sent it. Rick Klauser and I were competitors and collaborators, but it really was the same way.
Oh, that's always fantastic.
To the rest of the iron community.
What?
Similar to the rest, the majority of the iron.
Oh yeah,
community.
it's true.
Yeah, really.
It's just that it was so striking in a day of people starting companies and genes being worth money, that some of the collaborations in the mammalian endocytosis side weren't as robust as the iron people. The iron people were great.
Yeah, it's pretty amazing to me, having gotten to interview a lot of people like yourself, how it did seem like you really supported each other. And that's why, in the last 30 years, just an amazing characterization of iron homeostasis came about, which I'm really grateful for as a grad student, because I read all your papers. So it's very cool to hear different perspectives and how you work together. And it really did result in just incredible science that so many people benefit from. So
yeah, and it was, to be fair, it wasn't just iron. The copper people, we consider them our little brothers. They consider us their little brothers, but they were the same, exactly the same. And we in Utah benefited enormously from having Dennis Wingy down the hall, who really is an important copper guy.
No, that's
and
fantastic.
Dennis worked with Paul. Yeah.
Yep. Dennis is the reason I found Paul's lab. So yeah, and I started out as a copper person. So I just I love all metals, and they're all interesting and cool.
Yeah. And all the metal people I've dealt with are really wonderful.
Yeah, I agree. I would like to ask, just what perspective and advice you would have for younger scientists, both those interested in iron metabolism, as well as just generally, just any perspective you've had from your really incredible career.
So, taking out iron metabolism, I have two thoughts. Find something you really are interested and passionate about. That's part A. And part B, if 10 people are doing it, don't become the 11th. So a lot of people were working on virus membrane biogenesis. I was just one of a large number, and frankly, they were doing it better. So I went back to my first love, which is membrane turnover. How do cells turn over their plasma membranes. And it still interests me today. So find something that interests you. The other thing is, don't be terribly narrow. You can be narrow in what you do, but don't be intellectually narrow. Look at the whole field. You would be astounded where you learn stuff. I gave a talk at UC Davis, and a guy who I met talked about amphium red blood cell osmotic homeostasis. It was absolutely fascinating. So I came back and I started looking at osmotic homeostasis in alveolar macrophages. And we made a really cool discovery that we could synchronize both endocytosis and vesicle movement by messing around with different ions. And I think that's what Diane did for her PhD thesis.
It is.
It was great. I mean, it's still a wonderful story. Yeah. So, don't be afraid to learn new things. I think the way you do that is you go to scientific lectures. I mean, a good lecture speaker can introduce you to a field in an hour. Right? A bad one will put you to sleep, but a good one, I mean, that's how you learn stuff.
On that note, I'm going to make one comment about going to those science lectures. Jerry's the only guy I know who could go to a lecture, sleep through 3/4 of it. I think he was asleep, but ask the most pointed and important question in the whole seminar, or give insight as to how the experiment could be done and what they needed to do. And I swear, and everybody around us would swear, that he was sleeping through that part. So it always blew me away.
Diane is being very, very gracious.
Not, I think that would still happen. I watched it for 40 years. I know it's true.
So here's the other thing. My other advice to a young scientist: learn how to write. I was naive, and I said, I'm going to be a scientist. I don't know how to write. I need to learn how to write. In fact, just the opposite. A lot of what you're going to do if you want to be a productive science involves writing. And my old department chair back in Connecticut said to me, reading makes the educated person, writing makes the precise person. So learning how to write is just critical. Find yourself the best editor you can. Even if he beats you up, you'll learn something. The last thing is, when it comes to writing and reviewing stuff, lose your ego. I always told my students and postdocs, talk to other people. I don't have the keys to the kingdom. You'll learn something from talking to other people. And when I wrote a grant, I insisted that everybody in the lab read it, including those people for which English was not their first language. And as time went on, Liang Tao really enjoyed finding my grammatical errors. And they always add something to the grant. So even though so I would write a grant, I would give it to everybody to read. Everybody would read it. Dennis would read it. Diane would read it. After they read it, I would give it to Jim Kushner, who would take my terribly written stuff and put it into poetry. So even though everybody in Utah knew I couldn't write an English sentence, NIH thought I wrote an iambic pentameter. So you gotta be egoless about this, right? So
we called it Kushnerizing the grant.
Yes.
Because it was incredible.
Yeah, he was just wonderful. And he gave the most wonderful lectures, because he was such a clear thinker, so smart. He was a hematologist. He reviewed every one of my yeast papers and yeast grants. He wasn't a yeast guy, but if he didn't understand something, it was my fault. So I really, and NIH for many years was very good to us because they were very broad. They've become much more focused, and I think it's to everybody's detriment. And I'm not sure they have the same appreciation of basic science they once did.
Well, that was a lot of really great advice. I'll always don't be intellectually narrow. I really like that tidbit there, but it was all great. I feel like I could sit and listen to you tell stories about discoveries in iron metabolism all day. But I don't know, Diane, did you have anything else that you wanted
I'm
to
sorry.
ask?
One last thing. Forgive me. Remember, I told you about those papers on the mutant P?
Yep.
This past year, a woman whose last name I think is Bulk, I wrote it down, identified the mutant gene. I wrote her a fan letter.
It's fantastic.
Yeah. She wrote me back. She had never gotten a fan letter before. She's a very accomplished geneticist. And it turns out she had worked as a postdoc. I didn't realize this until after I wrote the letter with Roland Lill, and she had published papers with Dennis Winge. So it's really a small world.
I
That's
didn't
fantastic.
appreciate that Roland got trained in Bill Wickner's lab.
Oh, I knew that.
I
Yeah.
did not know that.
Yeah, in fact, that's how he met Janet Shore, and they were friends. Yeah.
Yeah, exactly.
Yeah.
Okay, I mean, there's lots of stories we could tell, and maybe we do a second round of interviews. But Jerry, this has been great. Thank you so much for taking the time to talk to us,
Oh, it's been my pleasure.
share stories of others.
Yeah.
And this has been recorded, and we will let you know when it gets put online for anybody to go look for and watch. And I look forward to recommendations for others that we might interview to add to the giants in iron.
Sure.
Yeah.
Thank you. Thanks a lot.
Yeah, and Cade, thanks for doing the interview, and everybody have a great day.
Can you stay on, Diane?